Thank you for taking time to see my research presentation. I'm a geneticist at the University of Texas at Arlington, and I'm interested in uncovering the molecular underpinnings of traits that occur in nature and medically relevant conditions. I use domestic pigeons for my research because among the thousands of breeds of pigeons, they have a diversity of traits ranging from behavior, physiology, color, shape, and form, and many of these resemble what occurs in nature or medically relevant conditions. I specifically chose to work with colors because many of these are known to be Mendelian, that is, you inherit one copy from mom and another from dad, and the interaction of these two copies is what determines your trait manifestation. In using this approach and small pedigrees and small sample sizes, I have achieved very high statistical significance of association, but this was possible only because and if the traits are correctly identified and similar traits but with different genetic bases are excluded from analysis completely, and if the correct gene is selected a priori. Multiple genes tested results in a loss of statistical power. One thing we know from vertebrate genetic studies is that genetic pathways tend to be conserved with the same pathway or components of the pathway causing similar traits in multiple other species. For example, loss of function of the enzyme tyrosinase results in albinism in several vertebrates and results in albinism in domestic pigeons. We have shown that the exon 3 is lost in pigeons. The related protein of tyrosinase causes brown in several vertebrates and a type of albinism in humans. And I tested this gene against the brown pigeon and an allele which is an albino form called Ashred, and again with very high significance showed a gain of function mutation in Ashred and loss of function mutations in brown. This result was published in Domian et al. In Current Biology. It was published in collaboration, but the experiments were done independently using separate samples and approaches. We also published a second finding. The protein SLC45A2 is a solute carrier responsible for transporting the tyrosinase enzyme and maintaining an ideal environment for its function. It is most notably evoked by the white tiger phenotype, and we have shown that it underlies plumage color dilution in this allele of the locus. The lightening is mild, but there are more severe alleles as well. But this pattern of conservation does not always hold true. For example, here you see the melanocyte membrane, and on the melanocyte membrane you have a gene, a protein called MC1R, the melanocortin 1 receptor, and it directs pigment synthesis toward black pigment or red pigment called eumelanin and pheomelanin respectively. However, when it is hyperactivated, it causes black pigmentation, and underactivated, it causes red pigmentation. It also interacts with other proteins, and when alpha MSH is hyperactivated or agouti is inactivated, it will also cause black pigment. Similarly, alpha MSH is inactivated and agouti is hyperactivated, you will get red pigment. What this means is the correct trait must be identified and excluded from other similar traits, because you can have multiple blacks and multiple reds. What we showed is that loss of function of MC1R in pigeons does not cause red, as it does in mammals. However, activating mutations of the protein do cause darkening of the plumage and of the skin. A novel discovery we made was of the gene melan-A, which stands for melanoma antigen recognized by T cells. And it was not known prior to this to be involved in pigmentation, except from one knockout study. It is, however, involved in melanomas. What we have shown is that mutations of melan-A do result in sexual dimorphism, so the males and females look quite different over here. And it also causes a type of variegation that is very similar to vitiligo in humans. However, this trait in domestic pigeons is a prized trait and is selected. These are some of my summary findings. I have excluded a lot of the statistical details and experimental design. However, I would like to thank our funding from the university and Dr. Fondon, interns and breeders who have helped us during this work. Thank you.